All right, so I'm going to talk, yes, as James was saying, a bit about the basics with HGVS. So this could be a little bit boring for people who are very familiar with HGVS, but actually, as I was putting this together, there was a few things that I actually learned that I hadn't been aware of. But then Andy will get into more of the interesting parts of, okay, what do you do if you're looking at a paper and you need to now recreate an HGVS expression and you might be missing some of the components to really put that all together. So hopefully the two talks will go together nicely as far as kind of making a whole picture of this. So shown here just a screenshot with the URL from the HGVS website, because they actually really have tons of great examples for all the different scenarios you can think of. Even within each section at the bottom of each page, they have a question and answers part, which actually is really informative as well. As I was looking at it, sometimes the questions that would come to my mind as I was looking at the section is then answered under the questions and answers where they give examples. So it's really helpful. Additionally, I noticed in the educational materials button on the website, they actually have Johan Den Dunham, who runs HGVS, has like a PDF PowerPoint that goes through a lot of the basics of HGVS as well. So that's a helpful tool in case anyone's looking for additional information as well. But all right, I'm going to jump into now the basics. So one of the main things that sometimes people forget about, that when we talk about HGVS, you really need to include a reference sequence because otherwise you don't know the context for, what cDNA change you're talking about. So I added an example here of, if you just say, oh, BRCA1 C.43668 to G, because there's different transcripts of BRCA, they're actually, this could mean two different variants that map to two different genomic coordinates. And so again, if you're just sort of only saying the gene and a C dot, that really is not enough information. Often this is how we, talk about a variant because we sort of assume someone knows what transcript we're talking about and everything. But again, when you're really presenting the full HGVS expression, you do need to include the transcript or, the whatever your reference sequence is to avoid issues like this where there's some ambiguity in what variant you're actually talking about. Another important point is the reference sequence must contain the variant residue that you're describing, meaning a coding DNA reference sequence actually does not contain introns and therefore can't be used to describe intronic variants. So even though we often express intronic variants this way, technically that's not correct because the position 357 plus 1 G is not actually within this NM sequence. So it gets a little bit tricky at times for how to represent these. On the website, they suggested giving it in the context of an NG and then talking about the NM. So even though we often still use this top one that says not correct, technically it is not the right way to do it. And for those of you who have ever submitted to ClinVar, you've noticed that even if you are submitting only on the NM on RefSeq, ClinVar still always asks for what genome build you're using because of instances like this where the intron, is not actually part of that NM, so they need to validate if the position you're describing is real. So I included a screenshot of a intronic variant in TNNI3 here, and you can see that the way it's annotated right now in ClinVar is that it's a no change, that at, you're just saying at 373 minus 10 is a G. So you'd wonder, okay, why did anyone even think to submit this to ClinVar? And it's because in the previous builds, the reference was actually an A, and the C was the reference, but that's on the C was the variant, but this is on the negative strand. So we used to describe this variant as a T to G, whereas now you maybe would describe it just as a G. So if you're not telling ClinVar what genome you're using for talking about this intronic position, they aren't able to validate whether that expression is correct or not. So it sort of shows the importance that, again, when we're talking about intronic variants, we need to provide more context than just the NM because of issues like this. A few other basics as far as reference sequence goes.
Steven, sorry, this is Deb. I don't know if you want us to ask questions now or later.
Oh yeah, now it's fine.
So a lot of papers don't have a reference sequence. Is there a way to use HGVS with an ambiguous reference, not reference, but an ambiguous transcript? You don't know the person trying to make that HGVS doesn't know the transcript because it wasn't given to them in the paper? Or can you just not use HGVS?
So that's something that Andy's going to talk about, a way to do that with the allele registry, that they have that option that sort of would help you determine across all the transcripts for that gene, this expression, is it valid on any of them? The one issue with that is sometimes it is valid on 2 different transcripts, sort of like that BRCA1 example I showed. And so I had this a few times where I've helped labs who want to submit to ClinVar where they've sent me variants like that, or I can't tell the transcript, and I've had to send it back to them and just say, because I'm unsure, I just don't think we should submit this to ClinVar because I don't want to put it on the wrong one and then have someone make a wrong conclusion about it. So it's better to not have it out there. But yeah, so it kind of depends on, if you're able to make a very confident guess about the transcript, whereas if you can't at all, it is difficult then to share that knowledge, and you don't want someone to misinterpret it.
Okay. All right. Thanks.
So on the HGVS website, they listed all the different approved reference sequence types you could use, so NCs, LRs, NGs, NMs. It was interesting that on the website, there was a note that they are still looking at if using ensemble transcripts is okay for HGVS, which I assumed it was because people use that all the time. But there was a question of that ensemble transcripts sometimes are not versioned, and you need to be able to include a version number when you're talking about a reference sequence. So I'm curious if that is a discussion that's still underway within HGVS about using ensemble transcripts, again, because so many people seem to use them when using HGVS anyway. So I'm guessing it's okay, but it looks like they have not officially declared that as a acceptable reference sequence. And then depending on the reference sequence you used, when you're describing your change, you would use different letter to indicate what type of sequence you're looking at. So a C dot for coding, a G dot for genomic, M for mitochondrial, and so on. That whatever letter you're selecting here correlates to whatever your reference sequence is that you're using as well. A few other basic points. They noted that all variants should be described at the DNA level, and that's because if you're just saying a protein change, you could have multiple DNA changes that give you the same protein change. So only using a protein change isn't appropriate unless you did some sort of protein mass spec and you've only found a change, that approach, as opposed to sequencing. But if you have done sequencing, you should be giving the DNA expression, and then you could add on a protein or RNA expression sort of in addition to the DNA change. Additionally, the HGVS calls out the difference between when a change is experimentally determined versus deduced, and that's whether or not they use parentheses to show those two differences. So technically, whenever we're talking about, we're saying this cDNA change goes to this protein change, you're supposed to use parentheses after the P dot to indicate we have never experimentally proven that this protein change is actually what's happening. We are just predicted that, this amino acid at 24 position has changed, whereas if you have confirmed via like RNA-seq, protein sequencing or something, then you could show this protein change but without the parentheses to show that you actually have proven that this amino acid change is actually happening and not just something that you are assuming is happening at the protein level. A few more other, general information is this information about prioritization, that if you have a change that you think could be sort of falls under multiple categories, so this happens a lot with something that's a duplication and an insertion, is you're supposed to work in this prioritization level of how you would describe it. So if something could be described as a duplication and an insertion, you would call it a duplication. If something could be an inversion and a duplication, you would call it an inversion. And so you sort of work down this order as far as deciding what is the preferred way to describe the change that you're seeing. And then additionally, depending on which type you're talking about, DNA versus RNA versus protein, the way that you call out the change varies. with the DNA, you're using, of course, the numbers followed by the nucleotides. And since it's DNA, you are supposed to use capital letters to describe the nucleotides, whereas at the RNA level, you use lowercase to describe what the ref and alt RNA nucleotides are. At the protein level, you can use the 3 or 1 letter abbreviation for the protein levels, but on the HGV website, they do say that they prefer doing the 3-letter amino acid because it's a little more clear than the 1 letter. You know, sometimes when you see the L, there's the second, we're like, wait, is that leucine or lysine? And it's just a little more thorough to put the 3-letter abbreviation versus the 1, but it's not incorrect to use the 1-letter version. All right. Sorry, screen froze here. Okay, so now going into some examples, starting with looking at cDNA. So this image at the bottom kind of shows you where you would start numbering based on where in the gene you are. So the numbering starts with the nucleotide 1 is the A of the ATG initiation codon. And then before that, anything that is before that A, the nucleotide right before it starts at -1, before that -2, and so on, which means there is no C.0. You would jump right from C.1, and the thing before is C.-1. So it's just something to remember as you're sort of going through numbering. And then the same is true at the 3' end. As soon as you stop, it then goes to a *1, *2. There is no *0 position when you're in the 3' UTR. And then for introns, you use the last bit coding position of the exon. And then if you are going into the 5' end of the intron, you're using the plus symbol to show how many nucleotides into the intron you're going. If you're talking about something on the 3' end of the intron, you use the first coding nucleotide of the exon and then add the minus sign for how many positions upstream you're going to. And as far as when you switch between plus and minus, from what I could see through HGVS, what you do is you look at the length of the intron and divide it in half. As soon as you reach that halfway point is when you would then switch from calling something positive such and such to negative something or other, sort of moving between the two positions. All right. So the most basic expression that people probably are the most familiar with is substitutions. Substitutions, we designate it with the little caret sign to show that a reference nucleotide has changed to an alternate nucleotide. However, we only use this caret when you're talking about DNA or RNA changes. So any of these examples here, at the protein level, we don't use the caret to show a change. Instead, you're listing what the original amino acid is, the position, and then what the new amino acid is. So this would not be 502 arg caret TRP, but instead you would use this format here.
I think I forgot last night.
So the next example is for deletions. So the format that they describe is that you use the positions deleted followed by the. Oh, sorry, if you don't have a question, if you could put yourself on mute.
I try to remind you, but sometimes it's hard. Sometimes it's forgot.
Excuse me. If you don't have a question, please can you put yourself on mute?
Okay, no problem. Okay,
let's keep on going. All right, so for deletions, I have some examples here of how you would use the nomenclature. So let's say, if you look at the sequence here and you found that you've deleted just this G. So you would call this, 2381 del G, or you can just call it 2380 del, because you're already describing a position within a reference sequence. It's not necessary to include what the nucleotide is. It does help, in the future if you're trying to, make sure it was a valid expression or something, including what the nucleotide is that was deleted can help, but it's not necessary. If, let's say, you have this example here where you're deleting these 3 nucleotides as GCA, you would describe it from the first deleted position to the last deleted position. And again, you can include the nucleotides or not. However, a invalid expression of this would of these 3 deleted would just be to give the first position and say deletion of 3. That would not be a valid HGVS expression because you need to give sort of the first and last position of where this deletion is occurring. Additionally, for all deletions and duplications, you should be describing these at the 3' most possible expression, and I'll show an example of this after going through deletions and duplications of what this whole 3' rule means, but it's something to keep in mind. So even in this case here, if these 2 G's, I would show that this G is deleted, not this one, because you would always show it to the most 3' expression possible, which again I'll show in a few slides. If you're talking about larger deletions, it gets a little tricky sometimes to figure out how you should be naming this deletion, and it depends on if you know the breakpoint and if you know if the surrounding nucleotides are included in it, or sorry, surrounding exons are included in a deletion or not. So let's say you don't know the breakpoints, but if you're looking, let's say, at this sequence here, if you know exons 30 and 31 are deleted, and you know that exons 29 and 32 are not included in the deletion, the way you would name this is you would start with, because you know the deletion essentially occurs somewhere in this intron and ends somewhere in this intron, you describe the boundaries of that intron then. So you're saying anything between 3190 plus 1 up to 3191 minus 1 is the boundary of where I know my deletion is starting, and I know it is ending somewhere in this intron, so anywhere between 3490 plus 1 up to 3491 minus 1. And that's how you would describe, again, this deletion where you don't know the breakpoints, but you know only it's including these 2 exons and not the adjacent exons. If you had this same deletion and you knew 30 and 31 were deleted, but you don't know the status of the surrounding ones, you would still include those inner boundaries to say, you're starting at 391 minus 1, 3490 plus 1, but then you would include a question mark on either side to show that you have not interrogated the status of the surrounding exons to know if they're included or not, so that you only know that the minimal part that is deleted would be within these coordinates. So jumping to duplications, it's a similar naming scheme as, oh yes, is there a question?
Yes, if the whole gene is deleted, do you put 1 or minus 1 as the starting point?
So I think actually, I should have included that. For those, if you know the full gene, I think on those they do it from where the beginning of the NM, so which could be like a C dot negative 100 or whatever the most 5' position of that gene is. So it's often before the ATG of the start. So you would use that, and then on the other end would show where the end of the 3' UTR is. And that's how you would say that's...
You may have tested that far. Maybe you have an MLPA kit, and you just have some probes in the exons. So you know the exons are deleted, but you don't know about the 5' UTR or the 3' UTR. So is it possible to just start with a question mark, and then 1, between question mark and 1?
So on the deletion page on the HGVS website, they do actually talk about the right way to do it for MLPAs, because you often are not, again, looking at all the exons, only some. But from there, I believe that they suggest that you base it on the location of the probe and not the boundary of the exon.
They say it's possible to put in the middle of the probe.
Yes, I think you would start with that, and then you would have and then you would use a question mark to show where it started, because you're unsure of how far out that deletion is.
Mm-hmm. But if you choose to just specify the first exon and the last one, do you have to use the first base of the exon, or do you put -1, -1?
I'm not quite sure. Because wouldn't you base it on where your probe is located and not the end of the exon?
I think both are possible. Maybe not.
Yeah, it gets very tricky with these large deletions as far as what is the right way, because there also was The nomenclature for this has changed a bit over the last few years, where it used to be you would use the first base of an exon, and then it looks like it changed to now where you do the plus or minus 1 to show that's the first position of the intron. But yeah, I'm not sure how, if that's happened all over or if it is just for certain examples or not. Yeah, sorry, so I'm not quite sure. Sorry about that.
Okay, no worries. Thank you.
So for duplications, it's a similar naming scheme that we saw for the deletions, where you're putting the first position affected and then the last. If you're doing a single position, though, you can just give the 1. However, you should describe anything that could be, any insertion that could be a duplication as a duplication, not insertion. So let's say in this case, if you inserted a G right here after position 2380, since the position right before it is a G, you would call this a duplication and not an insertion. So you would call it 2380 dup G or just 2380 dup, again, because you're already describing a position where the reference nucleotide is known. Whether you include that nucleotide or not is up to the person. But again, you would not call this 2380 to 2381 insertion of a G, because this would be seen as a duplication, not an insertion. If you had the case where you were now inserting 2 Gs into this position, you would now, because there are 2 Gs before this, you would now sort of expand it and say now it's a duplication of 2379 to 80 duplication of these GGs, as opposed to, again, saying that it's an actual insertion, because this matches the nucleotides leading up to this insertion of the 2 Gs. So it's now a duplication, not insertion. And for large duplications, again, sort of the same naming technique as the large deletions, where you would use, when you're known that the surrounding exons are not included, you describe it based on the beginning and end of the introns on either side, where the duplication would be occurring. So an interesting note that they put on the HGVS website is that in these scenarios, the duplication should be in tandem, but they don't say must be in tandem, which is interesting, because if you don't know it's in tandem, you also can't describe it as an insertion, because an insertion assumes you know where this duplicated piece ended up. So I think dup is sort of the initial way you would name it. If in the future you figure out exactly where it's going, then you could call it insertion if it's not tandem. But it's interesting, the naming doesn't really call out the difference from when it's confirmed tandem versus not. Although in those cases, I guess you would have sequenced the breakpoint, so then you should know. But yeah, it's just sort of an interesting note. It's always hard when they say a should as opposed to a must, because then it opens up to lots of different interpretations. All right. So for example of what we mean by using the most 3' expression, I've shown here the sequence for the CFTR delta 508 deletion. And you can see that you could get this deletion where this phenylalanine is gone from 2 different deletions, depending on if you're shifting one way or the other. So for example, if you're deleting the TCT, that is from starting at position 1520 to 1522, you can see that if you deleted this T, C, and T, these 2 T's from the phenylalanine would move on over here to complete this A, which would stay as a isoleucine, and then you would still get the G, GT, GTT for the glycine and valine. So you would just be missing that phenylalanine. However, you would get this same exact sequence if you instead deleted 1521 to 1523, which would be this CTT. So in this case, if you again deleted this C, this last base from the isoleucine, this T moves on over to complete this, so it stays again as isoleucine, just like in the other case, and then you get the glycine and valine afterwards. So the question becomes, okay, which one of these do you then use to describe this deletion? Because if you're just sequencing a patient and you get this sequence, you don't actually know which event really occurred. So HGVS has decided that this is where you would always use the most 3' representation. So in this case, because 1521 is downstream of a position that was started at 1520, we would use this expression to show that it's a CTT deletion as opposed to a TCT deletion, because again, you're getting the same result either way, so you'd want to use this approach. And this is something to definitely keep in mind with as you're looking at older papers, because there are times where, I'm curating a variant and maybe at first search I didn't find any publications with that variant, but then if you dig deeper, you realize there are some papers out there, but they did not shift, and so your searching may not have found the variant that you're actually looking in, because it could be, again, in a paper where they didn't right shift and kept it as a left prime representation for their variant. So one other thing to note about this that makes it even more confusing is, even though we're saying to use the right-shifted expression, if you are looking in any kind of genomic database like NOMAD or EXACT, since they use a VCF representation, which is always 5', your deleted nucleotide is not going to match what NOMAD is saying. So this is how this variant occurs within NOMAD, and they're using the TCT deletion as opposed to CTT, again, because it's VCF, which is 3' shifted. So it's something definitely to keep in mind that especially for genes on the positive strand, you're not going to see the exact same positions deleted. It's going to be shifted to the 5' most representation when you're looking in NOMAD or EXACT. One other sort of issue that comes up with this whole shifting is for genes that are read on the negative strand. The position you would describe with a genomic position and the position with the cDNA position are not talking about the same nucleotide because of these shifting expressions of going to the 3' most expression. So I have an example of that here because it's sort of an odd concept to think through. So if you think of the sequence here where you have the genomic sequence here going 5' to 3', and the cDNA is right on the minus strand, so it's now the cDNA numbering is going in the opposite direction. So let's say you have one of these G nucleotides deleted, which would be a C nucleotide at the cDNA level. So if you're talking about this deletion at the genomic level, you would be using the 3' most expression, so we would say that it is this G that is deleted, so we would call it G.5 del G to show that this one is deleted, and this position would correspond to actually what your C.4 position is. But if you were talking about this deletion at the cDNA level, so again we're going this direction, you would again be using the 3' most expression, which in this case is your C.7 deletion of a C, which corresponds to what your G.2 is. So you can see again, even though they're both talking about the same deletion, at the genomic and cDNA level, they're going to be implicating a different nucleotide within this string. And so this is something that's an issue as well within searching, that it's just something to keep in mind, because I've also noticed different databases shift this differently. I've noticed even within ClinVar and the allele registry, sometimes they call it the genomic position exactly what the cDNA position is, like they just map it without shifting it again. Sometimes they don't. So it's another sort of troublesome area for when you're looking at repetitive regions like this to really figure out if the deletion you're looking at and something you see within EXACT NOMAD or a publication really is talking about the same deletion that you're looking at. So hopefully that all made sense. I know it's a little bit of a complicated situation here. And then one exception to the 3' rule is when you are looking at adjacent nucleotides at the cDNA level that are actually not next to each other genomically because there is an intron between them. So, for example, even though 3921 and 3922 are both T's, if you saw deletion of this T, you would not shift it and call it 3922 deletion of a T, because you're actually able to confirm it was this T that's deleted because the surrounding sequences are different. So this is a case, again, where you would not do a 3' shifting, even though 3921 and 3922 are next to each other, because they're actually separated by an intron and not really next to each other. So you are able to interrogate which T really is deleted. All right, so moving on to insertions. It's, again, somewhat similar naming that we've seen with deletions and duplications, where you start with the flanking positions and then marking what the nucleotides are that are inserted. So one difference here with insertions versus deletions, duplications is you need to note what the nucleotides are that are inserted, because if you're just saying, so let's say this example down here, if you're just saying between 2380 and 2381, I'm inserting 2 nucleotides and not telling you what, you can't really figure out what the downstream consequence of inserting these 2 nucleotides are, because you haven't told me what's being inserted, just 2 nucleotides are. So you have to actually include what nucleotides are here so that you can, again, figure out what the actual consequence is going to be. Additionally, you need to give the flanking regions, so you couldn't just say 2380 insertion of a TA. You need to say between 2380 and 2381, I'm inserting a T and an A, so that you actually, again, are able to confirm where it's really going in, because if you're kind of just giving this one position, it gets a little unclear if did you insert it at 79 and now you're saying 80 is this, so that's why it's more clear to give the start and the 2 flanking nucleotides of the insertion, so to get rid of any ambiguity around what the what's actually occurring. So another interesting variant type that comes up with HGVS is repetitive regions. So I've shown an example here where you're seeing 6 repeats of a CAG. So the way that you would describe repetitive regions like this is that you use the nucleotide of the first nucleotide of the first repetitive unit, and then list the nucleotides and then a bracket and how many of those occur. So in this case, we'd be talking about C.52, CAG, bracket 6 to show starting at position 52, I then have 6 CAG repeats. Another important thing to note here is we are talking about the first repeat, not the 3' most repeat, because I just like to make things complicated. There's always exceptions to things. So for Del's duplications, we're using 3', but when you're talking about a repetitive unit, you're talking about the very first position of where the repeat starts and not the last position. One last thing to sort of go through is alleles of how to describe when you are seeing multiple changes within a person. So they use brackets to talk about a specific allele, and then a semicolon separates different alleles. So if you are seeing 2 changes that are on 2 different alleles, that's where you would do C dot and then bracket to describe what's happening on one copy of the gene, semicolon, and then new brackets to describe what is happening on the other copy of the gene. If instead you wanted to describe one allele that had several changes, so like a haplotype, that's where you would do C dot open bracket and then describe each one of the changes with a closed bracket and putting a semicolon between each one of these that designate all of these are happening on the same copy of the allele that you're looking at, and these are not in trans. These would be all in cis. Lastly, if you have multiple changes and you don't know if what you're looking at is in trans or in cis, that's where you put parentheses around the semicolon to indicate I am unsure of if these are in cis or trans, but I did identify these 2 variants in the person, and I'm unable to determine, which on which copy is each one of these occurring. Maybe they're in cis, maybe they're in trans. And then I added a few last things about protein nomenclature, because I spent most of the time going through DNA, because I feel like that's where it gets a little bit trickier. Protein nomenclature is a little more straightforward. Again, the 3-letter amino acid code is preferred. You can always describe things to the 3' or C-terminal, most position possible. Whenever you're looking at a variant that is impacting the initiation start site, so M1, you would describe it as MET1 or MET1 question mark, as opposed to putting in what the substitution at that position would be. And when you're talking about a nonsense change, you would use TER or the little asterisk button to designate a nonsense change. I think the asterisk gets tricky because that's also what people often use as a wildcard, so searching for that gets a bit difficult. And some labs use X. I work in a lab that actually uses X instead of these as well, which actually is not, technically does not follow HGVS standards to use the X. You are supposed to use the TER or the little asterisk button or symbol. And then here are just some examples of different protein nomenclatures that you would see based on, again, substitutions, nonsense, duplications in frame versus out of frame. For frame shifts, there are sort of two different ways you can represent the frame shift. You can do a short approach, which is just the position, or sorry, the amino acid position followed by a frame shift, just to say this is now going to be out of frame after this, versus a longer one where you are describing what actual change is happening at this position and how many new amino acids downstream would you see now a stop site. So in this case, you are now going to see a stop site 23 amino acids after your 97. So at position 120 is where you would see the stop symbol. So one thing I noted, because as I was, right before I started, realized one thing that's missing here is sort of a combination of frame shift, no stop, where you're seeing an extension. That often gets missed from different predictors giving you protein expressions, that there are times where you're going to have a frame shift and the new stop is actually further downstream than the native stop. And so I didn't have time to add in the example of what that looks like, but it's similar here where you're showing it's an extension and then where the new stop is and then how far downstream it is, to sort of call out the difference between when it's a frame shift and it's now a premature stop versus a frame shift that actually is extending the protein. Because if you're just using this expression, it's not actually clear if that new stop, where that new stop is relative to what the native stop position actually would be. And the last thing I just wanted to acknowledge Hannah, Zook, and Chrissy often see, who gave me many of these slides because they've taught this at a course here previous years, and so they were able to give me many of these slides. And I'm happy to take any questions about HGVS, or if we want to move to Andy's and do questions at the end, that's fine as well.
Thank you so much, Stephen. That was fantastic. Does anyone have any quick questions for Stephen?
I did have a question, and I think it was more speaking to, and thank you, Stephen, for this great presentation. Do we know how many of these actual HGVS nomenclatures are supported by our ClinGen allele registry? I know that on some of them with ClinVar, but are we able to use all of these different HGVS nomenclatures to return results?
So I believe the major limitation right now with the allele registry is unknown breakpoints. That also is a difficult issue with ClinVar as well. So let's say, I'm going to go to where I have one of those. So I believe right now, my computer freezes on this every time. So let's say you had this unknown breakpoint deletion here. If lab 1 submitted this deletion to ClinVar and then lab 2 submitted with the exact same nomenclature, I believe ClinVar actually does not aggregate those deletions because they can't be confident that you're actually talking about the same variant, because it could be that they have different breakpoints, and so since there's uncertainty, they don't put those together. And so I think that's why there's an issue with these within the allele registry as well, as far as how would you give a registry ID to a deletion like this versus, if someone else has also named it like this, but it's not actually the same variant, giving it the same variation ID, it can confuse things downstream. So I think that's the main one, is the uncertainty, and I think the only other limitation is that due to size, the allele registry, it has to be like smaller than 10 megabases, which there's not that many variants that are that much larger, so it's not that big of an issue. But yeah, I think the unknown breakpoints is more of the issue right now.
And also, my second question is there guidance around how to submit trinucleotide repeats?
Yes. So there's the so the one slide of it where it gets, there are a way where you can also use the parentheses to describe what you arrange. Like if I don't know the exact number, but I know it's between 100 and 110. that you can sort of call that out. And the HGVS page for the repeats actually has a lot of really great examples about how to do that and actually how to mix multiple types together. So like the TGT repeats in CFTR, of how you put that as one expression as opposed to having to call those two out separately. So I think that's sort of probably one of the most helpful ways to look at it is going to the website for those examples.
Okay, thank you, Stephen.
Great, thanks. So let's move on to Andy's presentation, and if there are additional questions for Stephen, we can take those at the end. But thank you so much again, Stephen. That was really terrific. So Andy, I'm passing this on to you.
Okay, great, thank you. Let's see if I can share my screen. Okay. Okay. So, as Jenny mentioned earlier, I have a quick presentation about some examples and some tools and hopefully some helpful hints on how to map some of the variants that have been published or deposited into databases earlier on, before HGVS or before the authors or submitters were aware of HGVS, so that they can be utilized for curations, either gene or variant. So just a quick overview of what we're going to talk about. So one of the common issues, as I mentioned, is that a lot of these variants are from older submissions or publications, and I've actually been doing a project that's been looking at a lot of the data in OMIM that's been submitted earlier on, and so they don't have a transcript associated with them, or they don't have the correct cDNA associated with them. But then some of the other issues that we run across is that no transcript is reported in the paper, and I know that was a question earlier, so hopefully I'll be able to help with that. The allele registry is very useful in this endeavor because they can map the variant using just a C dot or a P dot and show you all the different transcript impacts that you might have. And then they also have a tool where you can interactively generate a variant using one of the transcripts that they have in a window, and I'll show that as well. So as I mentioned, I've been working on a project involving a lot of the OMIM data. So this is actually part of an export from ClinVar of all the different variants that don't have HGVS expressions associated with them. So on the left, we have what they're called in ClinVar, and then what gene they're in. And then we can use the variation ID and the information on the variant page to sort of find the OMIM submission, which is linked to the paper that then describes the variant. And we can look at the paper as well as information surrounding the gene and genome browsers to elucidate a NM HGVS expression. And then I put this into the allele registry, and it can tell me if there's either a ClinVar entry that already has this associated with this variant or not, this HGVS expression associated with the variant or not, and then we can sort of eliminate this old submission, or if we need to make a new variation ID, or sorry, incorporate this new HGVS expression and overwrite this less useful old name of the variant. So this is what one of these variants looks like in ClinVar. The issue here is normally there's a nice HGVS expression that you can map. So as of right now with this variant, you could enter it into a gene or variant curation by using the variation ID, ClinVar variation ID, but this identifier is not necessarily universal, and it's not necessarily the most helpful, so it's good to have the HGVS expression. So let's get into some examples. So the first example of a lot of the issues that are from older papers is that a lot of authors used an old nomenclature that is IVS nomenclature, which is intronic variation sequence, used to describe splice site variants or intronic variants. And so an example of this is this SLC26A4 intron 4 variant that basically just reads, and I know many people may know this, but this is basically just saying that there's a to G change 7 base pairs from the downstream end of exon 4. And so what I typically do is I go into a genome browser, either Alamut or UCSC, and you can sort of find a transcript. Fortunately for SLC26A4, there's only one transcript, so you don't have to sift through quite as many until you are sort of sure that this is the area that you should be looking at. And I usually find, intron 4 here, which is right after this exon, and I go to the +7 site, and I would sort of look at any other information that the paper is given. So fortunately, this paper actually had the Sanger trace of the area that the variant occurs in. And while they sort of use their own reference sequence and call this position 639, we can see that with the transcript that is described in UCSC or Alamut, this position corresponds to this actual same position here with sort of the GGT, AAT, TTA, and we can sort of guess that this is the right point. And we can say, okay, so this is the transcript based on the transcript that is called an element. Sorry, I should have included this right here. But we can say, okay, so we have a +7 A to G site at this C.415 +7 in this transcript. And we can enter that into the ClinGen allele registry and maybe see if that other transcript that the paper used corresponds to this variant, to sort of assure us that this is the same variant. Unfortunately, this paper, actually it seems that this transcript was lost to follow-up, for lack of a better term, and they're sort of using their own transcript. But we can still, if we're interested in trying to curate this variant or associate it with a gene curation, we can take this expression, take the allele registry ID, and curate the information. Another one of the common issues is that a lot of papers just sort of describe what the variant is, and they give some indication of where the variant occurs, but they don't necessarily give a transcript or a indication of what's actually deleted. So in this example, we have this PYCR1 22 base pair deletion that's said to occur in nucleotide 617. So fortunately, this paper actually gave a little bit more information, and this is excerpted from OMIM, but they said that the deletion is at C.617, 633 +6, and it's a deletion, and it encompasses the exon-intron boundary of exon 5 in the gene. So what I did was I went into Alamut and I looked at the transcripts. You could also do this in UCSC. So there's actually 2 different transcripts. And this first transcript, if you look at exon 5, you can see that it actually ends, we're looking for at the +6 site. So we're looking for it to end at this 617, C.617. And this actually ends at the C.621 nucleotide. So we know that it's not this paper, whatever transcript it's referring to, it's not this transcript. So then I usually go to the next transcript, and you can see that this is actually, since this is the beginning of exon 6, that this end of this exon is actually exon, or the nucleotide 633. So then we can select this transcript, and we can take a closer look, and we can say, okay, so where does this deletion start? Where does it end? And if we're looking at this region that is said to be deleted, and we apply the right shift rule, the 3' rule that Stephen was mentioning, we actually get that the deletion occurs at C dot 618 to 633 plus 7, and it is in fact in that transcript. And then we can then put that into the allele registry to sort of validate it and look at some of the other impacts that it has on all the other transcripts. Next example is what if they just give the C dot transition. So this was an older paper that described a transversion from of a at position 103, a C to a T. And the best way that I think to figure out what this variant is to just go straight to the allele registry and use the gene and variation-based query tool, where you can enter in information about the variant in the gene, and it will sort of return results on all the transcripts that are currently registered in the allele registry. So if we put in our gene name, that it's a C dot or a P dot if we're looking at the protein consequence, which we'll look in a second, the nucleotide position, C and a T, and then we press the query button, we'll get all the different transcripts that this variant might appear on. And you'll notice that these are actually all the same allele ID because they all correspond to the same variant being mapped across multiple transcripts. So we can select any of the transcripts really, but if we want to look at the actual allele ID page, we can click on this right here, and it will bring us to the allele page, and we can see what the ClinVar variation ID is, the RCV information. Sometimes it'll pull in information about nomad if the variant is found in nomad, as well as the genomic sequence.
And Andy, I have a quick question.
Yes.
If you go back one, or maybe this is going to be your next thing, if you were to get to this screen and it was multiple different variants, does it call it out in a way you notice, or do you just look at the identifier column and genomic to see that they're all the same? Like if there was one that was different to show that, you know, this does exist on a different transcript and it's not the same variant, how obvious is it?
Yeah, so I usually do a quick scan of the identifier column, and it'll have a different allele registry ID if it's a different variant. But that is a good point. I probably should look at this as well to make sure, although they should be the same.
Oh yeah, if the identifiers are the same, I just meant if it's off by one digit, if you're looking really quick, is there a way that you can easily tell this could actually be multiple different variants?
Right. No, they don't highlight anything.
Okay.
But the next example actually does have two different variants, so we'll see that. So the next other issue is if they only report the protein consequence of the variant. So this LIPH variant is a transition from a cysteine to a serine at amino acid codon 246. So I went to the genomic browser and I pulled out the only transcript that's actually reported for this, but if there were multiple transcripts, I usually do a similar thing that I did for the first variant that we talked about, and I try to find the position they're talking about and see if it makes sense in the context of the variant. So I go to P dot 246 and I see if it's a cysteine. So in this case, this does make sense. The cysteine is at position 246. And there's a couple of different things that you can do if you can find the actual C dot. The simplest thing is to go to the allele registry and put, and I'll show this in a second, and put the protein and the P dot and the position and the change from cysteine to serine, and it'll map it for you. The other sort of fun thing that I like to do before the allele registry was able to do that is to look at the old-fashioned amino acid codon chart and look at the codons that generate a cysteine, codons that generate a serine, and find one that makes sense. So in this case, I would see that this TGT corresponds to or can be converted by one SNP to a AGT, and so we could say position 736 T to A in this transcript that we identified is a possible HGVS expression for this variant. However, it's probably better to go into the allele registry and do the gene and variation-based query and enter your information, and then return the results for all of the different possible transcripts, at least the allele registry knows about, that this protein consequence may have occurred from. So this is actually two different variants. As you can see, the identifiers are different. The nucleotide changes are different, and it's basically what I miss is that there's two different ways to generate the same amino acid because the genomic code is redundant. And depending on your situation and sort of how confident you need to be, you can pick one of these. Typically, if I'm trying to curate sort of the protein consequence itself for a gene curation, I'll just go with, that this variant's found in ClinVar and NOMAD, so this is probably the one that the paper was talking about. But again, you have to sort of use your judgment and decide whether you're confident enough in whether this is the actual change you're looking at. It looks like we have 2 minutes left, so I'll go quickly. So there are a few different other ways you can identify variants using the allele registry. You can use exact IDs, NOMAD IDs, dbSNP IDs. They also recently implemented a feature where you can enter a PubMed ID, and it might return variants that are in the PubMed publication, but this is sort of new and not all of the information in PubMed is uploaded to the allele registry, so hopefully more information will be incorporated soon. And then, as I mentioned quickly, the other way you can identify variants to interactively generate it. So if you click on this box right here and you enter in your gene name, you pick a reference sequence. We would pick this transcript because this is the most relevant transcript for USH2A because it's the long transcript. You can sort of look at this text box of the entire transcript and highlight the region you want to change, and it will tell you sort of where it starts and ends in the position you highlighted. And you can either enter, say, like I want to delete this and enter something else, or I just want to delete this. You can either put something in the alternate allele box or not, and it will return a result for that allele, whatever it is, and however it maps to other transcripts. If the variant's not in the allele registry, you'll get this notification right here, and if you click that, it will generate a new allele ID for you, and you can use that for your curation. So if you can't, for whatever reason, use some of these tools to get an allele ID, sometimes what you have to do is, in the VCI, you're kind of out of luck because there's no other way to identify it. Hopefully there's a ClinVar ID that you can use to at least generate an interpretation, even though it only returns transcript information in the VCI for SNVs. But for gene curation, what we typically do when it's a whole gene deletion with fuzzy ends or what have you is we make a note in the final summary and say, there was this deletion, we couldn't map it, but it was shown to cause NMD of the protein or what have you, and we would therefore give this the full points or decrease points for the curation or whatever you're doing. So in summary, as long as you can identify a transcript and you can sort of identify the region that your variant occurs in, you can hopefully write an HGVS expression and then verify it with the allele registry, and it will hopefully help you sort of sift through any right shifting or other issues that may come up with that variant. And then sometimes for the complex indels,